In Chapter 7, we will learn about the complement system. Take a moment and note how this is spelled, Complement. The complement, as in, oh, I like your new dress, that word is spelled Compliment. So they're two different words spelled differently. When it's spelled with an E, we're talking about the complement system of the human body. The word literally means to complete something or to make something whole. And so as we go through this information, kind of be thinking about why they might have named this system complement when it was initially discovered. Think in terms of what this system does. As we proceed through this material, some of the general topics that we're going to examine include the pathways of the complement system, controls on the system, biological manifestations of complement activation, complement and disease states, complement deficiencies, and laboratory detection of abnormalities. A little bit of history here. Back in the 1800s, it was observed that serum from an immunized animal could lyse that strain of bacteria. So in other words, we have an animal, they're immune to organism X. If their serum is added to organism X lyses. They also discovered that if we heated that same serum to 56 degrees for 30 minutes, the lysis no longer occurred. So they decided that whatever this was, in terms of, chemicals or whatever substance it was heat labile, meaning heat destroyed it and took away its activity. They also took it another step further and determined that if this serum that could lyse a given bacterium was given to an unimmunized animal, then that second animal would now be immune. So, conclusion of all this, there's a heat labile substance that complements or helps antibodies in their lysing of bacteria. So now we know that complement is a set of around 30 proteins, some of which are soluble and some of which are cell-bound. They interact with one another to enhance host defense mechanisms against foreign cells. Now, don't spend any time, the 30 is inconsequential. It's just to, give you this mindset that there's a number of these components involved. Most of the plasma components of complement are synthesized in the liver. So if you've studied anatomy and physiology and you've studied liver physiology and you've learned that the functions of liver cells are just tremendously varied and important, one of those functions of the hepatocytes is to synthesize many of the complement proteins. Most of the protein molecules that are part of the complement system are zymogens, and that's simply a term that means they're inactive until an enzyme converts them into an active form. And the reason for the little graphic of the dominoes is this. Probably most of us have done this with dominoes in our childhood, or maybe you go online and you can see huge demonstrations of thousands of them. But the whole thing is you push the first domino over and then the first one falls and pushes the second one over, which falls and pushes the third one over, etc. This is a very good analogy for the complement cascade, meaning that one step, once accomplished, triggers the next step, and so on and so on. And if you've ever made your own string of dominoes, you know that if you get them too far apart or out of place a little bit, it'll stop. They'll fall up to a given point, and then it doesn't go the rest of the way because something is out of place. This is a very apt analogy too for what happens in the complement system. There are a number of disorders that individuals have that are associated with their complement system. Either they're missing a component altogether, or they have it in low numbers, or maybe they have it, but there's something malformed about it. And what happens with these individuals is the first part of their cascade may start, but as soon as the cascade gets to the missing domino or the domino that doesn't work, nothing happens from there on. And so the dominoes falling in sequence, good analogy for what goes on when the complement cascade is initiated. There are many outcomes when complement is activated. Pathogenic cells can be lysed, and we'll look at that in some detail in a moment. Pathogenic cells are opsonized or made more attractive to neutrophils, drawing them into the area. Metabolism and the clearance of immune complexes are enhanced. In other words, complement helps us get rid of immune system garbage that's formed. We see an increase in vascular permeability. The system recruits monocytes and neutrophils to the area. It triggers the secretion of immunoregulatory molecules, back to those cytokines that serve to amplify the overall immune response. They help clear out the apoptotic cells, the dead and dying cells. The system does interact with the coagulation pathway. It helps join the forces of the innate and the acquired immune system so that they work together. Some complement fragments have roles in the activation of B cells and antigen-presenting cells. And it helps enhance immunologic memory through interactions with those B cells and dendritic cells. This complex complement system has three ways to get it started. We talk about the three pathways of complement: the classical pathway, the lectin pathway, and the alternative pathway. And we'll look at all three of those in a little bit more detail. The bottom line is these are the three mechanisms to start the system, to get the ball rolling, so to speak. Once the system is activated, it's going to go to the same completion. So we can have multiple pathways coming into play at the same time, or there might be simply one pathway that's leading to the activation of the system. In any event, the end result is lysis of the invading cell. We're going to blow that guy up and get rid of him, because remember, what this is all about is our immune system fighting off those foreign bad guys that are trying to get in and cause infection. Of the three pathways, the classical pathway is most important when we are fighting an organism that we have previously been exposed to and previously been stimulated to make antibodies to. This is because the classical pathway requires antibodies bound to the cell surface in order to be initiated. Classical pathway divided into three segments: the recognition, activation, and then the formation of the membrane attack complex. Classical pathway requires only one IgM molecule, or at least two IgG molecules, to be initiated. IgM is more efficient than the IgG because it's so big. It's got all those multiple binding sites. So if one large IgM molecule binds to two adjacent antigenic determinants that can initiate this cascade, while at least two IgG molecules must attach in order for recognition to occur. Here we're looking at the recognition stage, the first stage or phase of the classical pathway. So we know that there have to be antibodies involved. So if we come over here to our graphic, here's the cell membrane of our pathogen. And in this case, we've got two molecules of IgG that have bound to that cell membrane. Because of that, this circulating molecule known as the C1 complex comes down, and C1 is made up of three components: C1q, C1r, and C1s. So this thing comes down, and C1q binds to the Fc end of these two adjacent antibody molecules. That chemical reaction then activates C1r and C1s. And the other thing we want to be aware of is that calcium is required. And so in order for an individual's complement cascade to function appropriately, they have to have adequate amount of calcium. Let's look at recognition again just a little bit more closely. So what we have occurring is that the C1q recognizes the Fc portion of two adjacent IgG molecules, or this could have been one IgM. At least two of these globular heads of the C1q, these things, okay, must be bound to start the pathway. When they bind, the zymogens, remember those are just simply molecules that go from inactive to active. The binding of C1q activates C1r and C1s. And we have C1s that is now activated. And what it has the ability to do is bind with two other substrates once it's activated. The two substrates that C1s can bind with are C4 and C2, and we're going to pick it up there on the next slide, but the takeaway here is once that C1 is activated, that's pretty much the end of what we consider to be the recognition stage. Now, before we jump into this, let me make kind of a general statement, is that when these zymogens go from inactive to active, what happens is they're cleaved. They're split into two pieces. And generally, one piece is the activation component for the next step in the pathway. And the other piece is said to just kind of float away into Nana land. And we'll look at this later. We'll see that these pieces that float away have great biologic effects, and we'll swing back around to them towards the end. But right now, we want to kind of just focus on what happens to those cleavage units that serve as the activators for the next step in the pathway. So on our drawing here, looks similar to the previous one. Here's our C1QRS. It's found its two antibodies. It's become activated. We have C1s. Oh, it acts on C4. It cleaves or splits C4 into C4a and C4b. C4a is going to float away and do its thing. Meanwhile, C4b is a very important part to stick around here, because what also happens after C1s is formed is that it cleaves C2. So C2 is cleaved into C2b, okay, which floats away, and C2a combines with the C4b. So this combined thing now, what is it made of? It's made of part of the C4 molecule and part of the C2 molecule. They're now active. Okay, they're active enzymes. They attach themselves to that pathogen cell membrane, and their C4b2A is their name, but their other name is C3 convertase. If you remember your chemistry, anything that ends in ase, always going to be an enzyme. So it's going to be a protein, means it catalyzes a chemical reaction. In this case, the name is telling us that it converts, okay, C3. C3 convertase is formed. What's the other name for C3 convertase? C4b2A. Where is it at? Attached to this cell membrane. The C3 convertase cleaves C3 molecules, okay, into C3A and 3B. So again, we have, the original inactive C3 is acted on by the C3 convertase, which splits it into two parts, A and B. So it's getting a little hairy here, but stick with me. You can follow along and digest this. Where we're at now, we had our C3 convertase was formed. It's attached to the pathogen's membrane. What does C3 convertase do? It splits C3 into two parts. C3A is going to float away and do something else. C3b is going to attach to the cell membrane. Once that C3b attaches to the membrane, and it's also going to join up with the C4-2A, we call it C5 convertase. Why? Because it's the enzyme that converts C5 into its active form. So now we're down here, our C4b2A3b, also known as C5 convertase, attached to the cell wall. It works on C5 to split C5 into C5a and C5b, and C5b is sitting here on the cell membrane. Now this C3b that floated away is an extremely potent opsonin, and what it's going to do is attach in multiple places around the outside of that cell. And remember, opsonins serve to attract the macrophage to facilitate the phagocytosis of the pathogen, get rid of him that way. Now we're to the final act here, and the final act is the formation of the membrane attack complex, MAC for short. So we started with our C5b, which is on the cell membrane, and then we go through this series of additions to this complex of C6, 7, and 8. And so 6, 7, and 8 come in here and they attach. And then the last scene is that multiple molecules of C9 form, and they stack together, so they form the cylindrical structure, and the middle of the structure will become a hole. So what we've effectively done is poked a hole in the cell membrane. And keep in mind that, one bacterial cell, when it's under attack, it may have hundreds and hundreds of these MACs that are forming on their membrane. And once we have that hole, then they're is an open channel for water and sodium to flow into the cell, and the cell is going to lyse. Now, I want to bring one thing to your attention because it relates to our case study this week, and that is this C8. C8 is an especially important one. I mean, they're all important. They're all required for the formation of the MAC. But C8 is really the one that when it combines in here to form this complex, it really embeds this thing. It really anchors it through the cell membrane so that all those pieces of C9 are firmly attached. Here's one more look at the formation of the membrane attack complex. Different artist rendition. I like this one because it's kind of three-dimensional looking. But anyway, C5 cleaved into 5A, which floats away. C5B converts C6, C7, 8, all come together to form this, this complex in the cell membrane. Then multiple C9s come in and align themselves in such a way so that when it's all done, there's a hole in the cell membrane, and the hole allows the contents of the microbe to leak out, and the microbe will burst. Now we'll look at the lectin pathway, also known as the mannose binding lectin pathway, or MBL. The lectin pathway does not require antibodies. Rather, this pathway is activated by recognizing carbohydrates on certain microbial cell walls. The mannose binding lectin itself is an acute phase protein. In other words, it's present when inflammation is going on. The MBL, the lectin, binds to mannose or other sugars, and it too requires calcium in order to bind. It's thought that the MBL pathway serves as a defense mechanism in infancy, in between the loss of maternal antibody and the development of a full antibody response. So within that first year of life, when mom's antibodies are diminishing, and it takes some time for baby's antibody productions to ramp up in the presence of certain microbes, this baby can still mount a complement response. The key thing is here, once C4 and C2 are cleaved, in other words, once we form C3 convertase, the pathway is identical. So the only thing that's different is the way that it is initiated. The alternative pathway, activated by the presence of bacterial or fungal cell walls, viruses, tumor cells, or some parasites. What I want you to remember about the alternative pathway fundamentally is that it requires some other factors. Factor B and factor D are part of the alternative pathway, as well as factor P, also known as properdin. Here's another look at the alternative pathway where we have microbe. We have C3 that's been activated by water. And so then we have the interplay between factor B and factor D. And attached to the microbe membrane then also is the molecule known as properdin. And when we're said and done, we end up with a molecule that has C3 convertase-like activity. And so once we get to that, then it proceeds to the membrane attack complex. Review of the alternative pathway, the key points, also known as the properdin pathway. It is part of innate immunity, but it bypasses C1, 4, and 2, doesn't wait for antibody to be there. for activation. And it's thought that once the classical pathway is initiated, this one kicks in and acts synergistically with the classical pathway. And here's the final take home comparing the three. We have the classical pathway activated by antibody molecules. We had the lectin pathway, which we learned appears to be very important in the first year of life, and the alternative pathway, which seems to be important in augmenting the classical pathway when it's activated. Now, they take some different steps here and take some different things to get them started, but the key thing is we need that C3. That's kind of the linchpin in the whole thing. And once we have C3 convertase, then we can make C5 convertase. And once C5 is cleaved, then we all go towards the membrane attack complex. Now, once the complement cascade is triggered, it goes very rapidly and it amplifies each step of the way. And so, in order to control this thing, the body has multiple controls in place. And there's a couple of them I want you to know by name. C1IH is C1 inhibitor. And one of our case studies deals with a patient who lacks C1INH. Well, his name is very descriptive. It stops C1 from becoming activated. And I liken it to the brake pedal on the car. It turns out that a small amount of complement is constantly trying to convert C1 into and cleave it. And so it's kind of like when you're in your car and you're sitting in a red light. And even though you're not pushing on the gas, if you take your foot off the brake, you'll creep forward a little bit. There's that little bit of creep. And so you have to have your foot on the brake if you really want to stay stopped at that red light. And so it seems that in our body, the C1 inhibitor is like the brake at the stoplight. It keeps complement from getting going on its own. Also be aware of factor I, and just take a look at these others. And now that you know a little bit about these various components and how this whole thing works, you can read through this carefully and kind of plug these things in and see how they affect the different pathways. It does seem, though, that in the end, most of these control proteins ultimately aimed at halting the accumulation of 3B. And back at the beginning, I pointed out that some of the complement proteins, those associated with the complement cascade, are not in the serum, but rather they're bound to cells. This is an example of some of the cellular complement controls. Now read through this. Don't spend time memorizing all of these. I don't think that's important at all, but I just want to give you the complete picture. And to me, you know, all these controls suggest to me that there's an awful lot of built-in brake pedals for this system, because if it's allowed to initiate and run unchecked, we've got a big problem. Which brings us to the association of complement and various disease states. So complement can definitely be harmful if it's not behaving appropriate or certain situations crop up. For example, the patient with gram-negative septicemia. All those gram-negative organisms circulating in their blood can trigger fulminant complement activation, and that activation of complement will actually contribute in many cases to death in these patients. If there's a lot of tissue necrosis occurring in myocardial infarction Or let's say someone has had a horrendous, let's say a car accident. There's massive amounts of tissue damage that is now undergoing necrosis. We have complement problems. When there are large-scale red cell lysis situations happening, that also can activate complement. And now that we are aware of all these different complement components, there's basically diseases associated with deficiencies of, I'm sure, any and all of them. These are diseases that it's only in recent years we can accurately diagnose and perhaps treat the resulting disorder appropriately. But read through these. What I want to point out specifically on this, remember we said C3 is kind of the linchpin of everything because we have to convert C3, which then activates and converts C5. People who don't have any C3 have severe recurring infections, and in fact, they may not survive the first portion of their life. And we do address that in one of the case studies. If a person is missing any of the components that make up the membrane attack complex, they tend to chronically be fighting infection with Neisseria because apparently the MAC is the body's #1 way to prevent us from developing Neisseria infections. We have a case study that deals with the patient with a C1 inhibitor. That's the hereditary angioedema case study, and on down the road. So take a look at these and in your own mind correlate what you know about that factor or that component in the complement system and how and why it might relate to some of these diseases that are associated with its deficiency. So what role does the lab play in detecting abnormalities in complement? Well, there's basically two categories of testing for complement. We can test for absolute numbers of the individual complement components in the patient's serum. This allows us to quantify how much C3 or C5 or whatever it is we assay for they have. But the problem is these methods don't determine whether that particular component is functional or not. So they may have adequate amounts, but what they have doesn't work quite right. So a couple of the methods include radial immunodiffusion and nephelometry. The other category of lab assays are assays that measure functional activity. And usually when running these, the end result is you're looking at lysis in the test system. Because remember, when complement goes to completion, we're going to have lysis of cells. And so in these systems, usually we use red cells in order to provide observable hemolysis. So there's an assay known as the CH50. This assays the classical pathway. There's a link on Blackboard for you to explore, learn a little bit more about this assay. There's an AH50 that tests for activity in the alternative pathway, and there's a number of ELISA methodologies out there. Here's another table I want you to independently spend some time with. What I don't want you to do is try to memorize this stuff. This is not the kind of thing I'm going to pin you down on an exam, but rather I want you to read through the table and try to rationalize, well, okay, if the patient is deficient in C1q, and or S, which pathway is that going to affect? Well, this says the lectin will be normal, the alternative will be normal. It's only the classical pathway that's adversely affected. That should make perfect sense to you at this point. So, like I said, read through it, use your brain, see if this all makes sense to you. This chart gives us a little bit different look at the components. This is just a classical pathway, and what those components are made up from or with, what happens to them when they split. And then lastly, I like several of these over here. For example, C3A mediates inflammation, is an aflatoxin. C4A mediates inflammation. C4B binds cell surfaces for opsonization. Again, please do not spend a lot of time memorizing this stuff. I will not ask you these specifics. I like this graphic because of its simplicity. Here's our three pathways: classical, alternative, lectin. Tells us what stimulates each pathway, whether it's an antibody or the surface of a pathogen. Tells us what's involved in getting to C3 convertase. Whether it's the factors B, D, and P in the alternative pathway, the lectin, and these others in the lectin pathway, etc. And then with C3 convertase, then splitting C5, then from there, everything is the same. We end up with the membrane attack complex as well as the opsonization that's brought about by C3b and the inflammation due to the pieces of C4, C3, and C5. To summarize, complement is a group of proteins that interact and in a very sequential manner. The activation of one component leads to the activation of the next. Very important in both the innate and the adaptive immune systems. These complement proteins are heat labile. Sometimes when we run lab tests, we are instructed to inactivate complement before we run the test. That's easy to do. All we need to do is heat that patient serum to 56 degrees, hold it there for about 30 minutes. All those complement proteins are going to denature, and there'll be no complement activity left in that patient's serum. The lectin and the alternative pathways are more important the first time we're infected with a new organism because no antibodies are required in order to get them started. Whole host of complement activities include the lysis of foreign cells, and we've talked primarily about pathogens, but I do want to put this in here because it is important. When we have self cells that are damaged and need to be removed, complement also helps us do that. Complement provides for the opsonization of cells. It results in increase in vascular permeability, leads to the attraction of monocytes and macrophages to areas where they are needed, helps clear out immune complexes that have been formed and now they need to be disposed of, helps clear out dead and dying apoptotic cells. It does have a segment that interacts and interplays with the coagulation pathway, blood clotting. It helps combine the forces of the innate and the acquired immune system. Some of these fragments have roles in the activation of B cells and antigen-presenting cells, so very important in especially the innate immune system, and especially important in the acquired immune system. It enhances memory through interactions with B cells and dendritic cells.